Okay, we're going to get back to the presentations now. Now it's my pleasure to introduce our next group. Vinod Handagama, Nitin Krishna, and Margaret McDaniel are going to talk to us about quantifying limits on replication, death, and quiescence of Mycobacterium tuberculosis in mice.
So, as many of you may or may not know, we initially started out as the Jonas disease group. So essentially what happened is on the first day, Dr. Ida and Dr. Ganusov gave us a paper as kind of like, oh, read this over, and these are kind of some of the techniques that you'll be using throughout the project. And when we came back, they were very, very intrigued by some of the very minute findings, and they essentially told us to spend a little more time with it, and a one-week assignment essentially became an 8-week project. So to watch the progression was very fun. So today we're going to talk to you guys a little bit about some background biology about the tuberculosis pathogen, and then we're going to discuss the models that we use and some of the conclusions that these models led us to. So tuberculosis is one of the oldest known human infectious agents, and it's second only to HIV/AIDS as the greatest global cause of death attributed to a single infectious agent. The incidence of tuberculosis is primarily in developing countries. Approximately 9 million new cases are diagnosed annually. These are active cases, but it's estimated that about 1/3 of the entire human population is actually infected with latent tuberculosis. So Mycobacterium tuberculosis is an acid-fast gram-positive bacteria, and what this essentially means is that it can't be stained and characterized with conventional staining techniques. And this is primarily due to their complex cell wall. Now, normal bacteria have peptidoglycan cell walls. These mycobacteria, actually have a very extensive array of lipids that they've attached to these peptidoglycan crosslinks. And as you can see, these colonies are very shiny appearances, and this is because very, very high concentrations of fats within their cell wall structure. And these fats contribute to the resistance to chemicals, the body's immune defenses, and allow these pathogens to actually persist on surfaces for long periods of time without drying out because of this fatty layer of their cell wall. So tuberculosis is transmitted primarily through droplet transmission. So what this means is that if an infected person with active tuberculosis coughs or sneezes, or any kind of fluid leaves their respiratory tract, if that is inhaled by a non-infected person, that person will become infected. And so what happens is the tuberculosis travels down into the lungs and eventually takes up residence in the alveoli. And the alveolar macrophages, which are primarily responsible for clearing any dust or particulate matter that would normally be inhaled by the person, identifies this pathogen as a foreign particle and then phagocytoses this pathogen. So at this point, one of two things can happen. In 10% of individuals, the disease progresses to an active state where the person will begin to exhibit symptoms of coughing, chest pain, bloody sputum, etc. But in 90 to 90% of cases, tuberculosis actually becomes latent. And the mechanisms behind this latency are still being elucidated by modern studies. And then at a later state, when the individual is immunocompromised due to old age or bad living conditions, this tuberculosis pathogen can awaken from dormancy and then cause an infection. Now, after this infection, in most individuals, or the majority of cases, the immune system of the host can actually clear this pathogen pretty readily. But in some cases, this pathogen isn't cleared from the individual's body. And there's still a lot of gray area as to why this is actually happening. We don't know if it's because the immune response isn't detecting the pathogen or if the pathogen is actively evading it, or that has yet to be elucidated. So there's been a lot of studies conducted with animal models, everything from zebrafish to rhesus monkeys. But one of the most popular infection models is actually the mouse model. And in the mouse model, it's popular because it quickly progresses to a chronic stage of infection where the bacteria counts, the population counts aren't changing very much. And there's two major hypotheses or categories of hypotheses to explain why the bacterial populations aren't varying. The first hypothesis is that these infectious bacteria that are actively replicating go into a state of dormancy or quiescence. In this state, they are not dividing, and they're essentially very low metabolic activity. Another hypothesis or set of hypotheses is has to do with dynamic equilibrium, saying that the reason the population counts are remaining stable is not because the bacteria are going into a non-replicative state, it's that they're actually being held at bay by the body's own immune systems. So to kind of further investigate these hypotheses, we looked at a paper by Gill et al. So what Gill et al. Did was they essentially devised a method to assess the replication dynamics of the tuberculosis pathogen in vitro. And what they did was they took this plasmid, this PB10 plasmid that carried kanamycin resistance, and they transformed the bacteria, Mycobacteria, with this plasmid so that they could differentiate these bacteria have plasmid, these bacteria don't have plasmid. Now this plasmid would be lost as the bacteria replicate in vitro. And based on the proportion lost during each replication cycle, they developed a model that could be used in vitro to correlate the proportion lost to a replication rate and a death rate. So as you can see here, the method that they used was they took in vitro cultures infected with the plasmid, and they would plate them onto two different plates. One of the plates had kanamycin, and the other plate was just normal media with no antibiotics whatsoever. Only the bacteria that contained the plasmid were able to grow on the kanamycin plates. So then they would take the ratio of the bacteria with the plasmid that grew on the kanamycin plates to the total number of bacteria that grew on the unspecialized plate. And then they term this the segregation rate, or essentially what percentage per replication cycle of the bacteria would lose their plasmid. And they found that the segregation rate was 0.8 throughout in vitro infection. So what this essentially means is if you have 100 bacteria with the plasmid initially, after a replication cycle, 18 of the bacteria would lose the plasmid, so 82 bacteria would retain the plasmid. And then they use this segregation rate to calculate the rates of replication and death using an in vitro model. They then sought to use this model to explain in vivo infections. So what they did was they aerosol infected mice, and over a period of 111 days, they would sacrifice 5 mice, and they would use this plating method to determine the bacterial colony counts. So this is their data. The black data points are the total population of bacteria, and the red data points are the population of bacteria that have the plasmid. Now, if you can remember from the previous couple slides ago, I talked about the two hypotheses, dormancy and the immune system keeping the bacteria at bay. So these are the two hypotheses to explain this flat line. The dormancy hypothesis states that the flat line exists because the bacteria aren't replicating anymore. They're going into a state of dormancy or quiescence. The dynamic equilibrium hypothesis says that the bacteria are being kept stable because the immune system is killing them at a rate that's roughly the same rate as that they're replicating. This data by Gill et al. Showed that the bacteria are losing the plasmid while in chronic infection. And the only way this could happen is if the bacteria were actively replicating. So Gill et al. Essentially used this data to support the hypothesis that bacteria are being held at bay by the immune system and they're not going into dormancy. So some of the objectives of this study when we were looking at this paper were we wanted to see, Gill et al. Held the segregation rate constant. We wanted to see what the effects on the rates of replication and death would be if we varied it over time. We also wanted to see what level of dormancy we could account for if we included dormancy into their model and see what the limits of dormancy would be. What percentage of bacteria are permissible to enter dormancy, and we can still explain the data. So now I'm going to turn it over to Nitin.
So in this talk, we'll discuss the development and analysis of 3 mathematical models that we've worked on for the past 8 weeks. We'll first discuss a general model, which is an extension of the model published by Gill et al. We'll next discuss a model that's also an extension to this general model that accounts for quiescence or dormancy of Mycobacterium tuberculosis. We'll next discuss a mechanistic model which accounts for macrophages, infected macrophages, and the immune response. So our general model follows the evolution of the total bacteria count, plasmid-containing bacteria, and plasmid-free bacteria over the course of 111 days. The equations are simplistically built in that they don't account for biological interactions other than replication rates and death rates. The difference between this model and the one published by Gill et al. Is that the segregation rate, S of t, was assumed to be constant in their model. Although this constant segregation rate was found to be true in the in vitro studies done by them, we don't necessarily know if this is true in vivo. No, the replication rates, death rates, and segregation rates are termed to be functions of time. So we take these to be piecewise constant functions defined by the intervals in which data were collected. So we define this as interval 1, then interval 2 right here, interval 3, interval 4. The replication, death, and segregation rates are assumed to be constant throughout those intervals, but could change between those intervals. Our parameter estimation techniques were adapted from Gill et al. They relate the 3 parameters with 2 equations. Here the replication and death rates are in terms of the slope ni and slope pi. Slope ni and slope pi are estimated from regression lines by fitting lines through the ith intervals and finding the slope through them. So slope ni would represent the slope of this line right here. Slope pi would represent the slopes here. The difference between the parameter estimation technique that Gill et al. Did and one we did is that this SI right here was assumed to be 0.18 for the in vitro studies and in vivo studies done by them. Since we do not know this is necessarily true, we just assume it's variable and solve parameters accordingly. So analysis we can do with this parameter estimation technique is one, we can assume S is constant throughout the infection, just like Gill et al. Assumed. However, since we don't know if it's 0.18, we'll test out different values for S throughout infection. So the graphs represented here show values for R and D that stay consistent with the data published by Gill et al. We find that as S increases, we require a lower replication and death rate throughout infection to still explain the data. This finds limits on the parameters. For example, we find that for an S value of 0.204, we reach a death rate of 0. And any probability of plasma loss greater than this would require a negative death rate to explain the data. Since negative death rates do not biologically make any sense, we say that any value of S between 0 and 0.204 is biologically meaningful. We also find that if death rate is constant throughout infection, any value for death rate less than 0.05 would correspond to an S value greater than 0.5. Since the value for S always has to be between 0 and 0.5 mathematically and biologically by definition of the segregation rate, we cannot explain the data with immortal bacteria. If the death rate is 0, we see that the value for S required to fit the data, it actually becomes 3.2. Therefore, bacteria must die throughout infection, otherwise we cannot explain the data. When we do the parameter curve fittings, we find that the values for rates of consecutive intervals don't look very different. So this suggests that the rates aren't actually different. So we do a bootstrapping analysis, whose algorithm is defined here, and we calculate p-values for the test that the differences are significant or not significant. And a p-value of less than 0.05 implies that there is a significant difference between the parameter rates. And the p-values are listed here. This implies that there is a significant difference between R1, R2, R2, R3, and D1 and D2. Therefore, we can refit the model, this time saying that constricting that R3 equals equal to R4, and D2 equals D3 equals D4. And when we do this fitting, as represented right here in the middle column, we get a lower AIC score than what was previously modeled. This implies that we can actually explain the data with only 3 replication rates and 2 death rates. We also find that we're able to explain the data if the first 2 replication rates right here are equal and the last 2 replication rates are equal and still with 2 death rates. This implies that we're able to explain the data with a constant replication rate throughout early stages of infection and then another constant rate throughout latter stages of infection.
Okay, so the next type of model that we're going to discuss is the quiescence model. And as you can see, the equations look very similar to the general model that we were just talking about. However, the difference here is that we have 2 new compartments, PQ and FQ, which are going to represent the plasmid-containing bacteria once they quiesce or go into dormancy, and the plasmid-free bacteria once they quiesce. Now, every replication cycle, Q% of the bacteria are going to be quiescent. And once they travel into the quiescence department, they are never going to replicate or die once they have reached quiescence. It's kind of like the immortal state. So with this model, we refit the data to see what types of levels of quiescence could be supported by the data. And we tested different levels of quiescence from 1% to 10%. And as you can see, some of them fit a lot better than others. What I want to point out is a particularly interesting fit from the quiescence level of 10%. As you can see, that pink line is kind of flatlining at chronic stages of infection. So that kind of confused us at first, like why are no bacteria dying? But really it's because at that point, because the quiescence is so high, all bacteria left in the system actually are quiescent and therefore cannot be killed. So judging from this result, we can say that a level of quiescence of 0.1 or 10% is unsupported by the data. However, what we do notice is that lower quiescence rates, like quiescence of 0.025, we can still explain the data very well with both replication and death rates within physiological conditions. So a quiescence rate of 2.5% per replication cycle might not sound like a lot initially, but at day 111, we actually find that about 33% of the total bacterial burden is going to be quiescent under these conditions. So that was the first type of quiescence that we tested, and we found that the maximum quiescence would be about 2.5%. The next type of quiescence we tested is that if the bacteria only quiesce at chronic stages of infection. And we refit the model similarly, and we also found that the maximum quiescence rate would be about 2.5% again. The next hypothesis that we tested was maybe whenever bacteria have quiesced and are then replated on the agar plates to count the colony formation, maybe those quiesced bacteria don't wake up, and so therefore would not be counted in the total bacterial burden. So we refit the data again, except this time, the total number of bacteria and plasmid-containing bacteria would not include the quiesced bacteria. Under those conditions, we could support a higher level of quiescence, up to 5% quiescence per replication cycle. So one thing that confused us a lot during the course of our research was that we found lower replication and death rates at chronic stages of infection. So generally what we would think about in chronic stages of infection is that your immune system would hopefully have a chance to identify the pathogen and start eliciting some kind of immune response to kill it. However, what we were finding is that the death rate had decreased, which we would expect the death rate to increase. So we decided that this meant we didn't really understand the underlying mechanisms of tuberculosis infection. So with this, the mechanistic model, we were aiming to try to understand what is actually happening during the infection, and hopefully then we could explain why we're seeing this decreased death rate. So with this model, what we are including is macrophages, bacteria, infected macrophages, and eventually the immune response. So like what Node was saying, in a tuberculosis infection, bacteria infect a macrophage, and these infected macrophages can either kill themselves by apoptosis, which is cell-programmed death, which is good for the body because it kills all the bacteria, or the bacteria can overwhelm the macrophage, and the macrophage will actually burst, releasing more bacteria into the system. So that's the basic idea of our mechanistic approach. We have the macrophages that are becoming infected at rate beta by bacteria, and then the infected macrophages can die by necrosis, which is delta, or apoptosis, which is alpha. If the cells die by necrosis, we see the bacterial count is increased at a burst size of n every time an infected cell is necrosed. Then those bacteria can then re-enter the system and infect more macrophages. We also added an immune response to the system, and what we noticed with data about the immune response is actually after a tuberculosis infection, the first 14 days we don't see any immune cell proliferation, and that's kind of still a confusing aspect. But what we do see is that from day 14 to 21, there is a great proliferation of immune response. So that's why our immune response is piecewise defined, and it will be increasing between the time intervals of 14 to 21. Whenever an immune response is affected and added to the system, it's going to increase the rate of apoptosis, and it's going to increase the refresh or the recruitment rate of other macrophages to the site of infection. One important thing to note about this model is that because, once again, we are modeling data of bacteria burden, one of the key parameters that's going to be affecting bacterial burden is necrosis, because that is how cells are releasing bacteria and increasing the bacterial population. So how we decided to model necrosis was not going to be a constant function, because as I said, necrosis generally happens whenever cells are overrun with bacteria. The bacterial burden is high, which would generally happen at later stages of chronic infection. So for that reason, we tried different types of functions to model necrosis. One of them was an exponential function, and another one was a logistic growth function. Both of these were done so because we wanted to have the elevated necrosis rate at the later stages of infection. We then fit the model to the data again. The top two graphs show when no immune response was incorporated into the equations, and we see that we're able to fit either the total number of bacteria or the plasmid-containing bacteria, but we're unable to fit both data sets simultaneously. So that was motivation for us to add an immune response to our model. We then added the immune response, and with a necrosis rate that was exponential, you see we're able to start getting the general trends that we are desiring for our fits. But whenever delta is logistic, we actually get pretty decent fits for both of the data sets, which is pretty exciting. So like I was saying, one of the things that was really confusing to us in our research was this decreased death rate at chronic stages of infection. So one of the mechanisms that is known about a tuberculosis infection is something called granuloma formation. So whenever a bacteria infects a macrophage, healthy macrophages are recruited to that site of infection, the idea being that hopefully somebody can kill the bacteria. However, tuberculosis is interesting because it's evolved in such a way that it's able to kind of take advantage of this interaction of cells, and that actually prevents itself from being killed by the macrophages. However, because now we have an aggregation of cells, it's kind of like the tuberculosis's defense mechanism now. It's going to be difficult for the immune response to penetrate this aggregation of cells. So what we wanted to do is we wanted to use a program called CompuCell 3D to try to model this aggregation of cells and to see what kind of conditions would have to be present in order for a granulomas formation to arise from the simulation. So what we did here is we constructed an aggregate of cells with the green cells being infected and the blue cells being healthy macrophages. We set the conditions so that infected macrophages would release a cytokine, and the bottom is just like a heat map of the concentration of cytokine. So as time progresses, the cells are releasing more cytokine, and it's attracting healthy macrophages to the site of infection. At later time steps, up to Monte Carlo step 3000, we see that all cells are actually starting to aggregate in a cluster. At further time steps, we would expect to see the infected cells aggregated in the center with healthy macrophages around the peripheral, which is a general idea of what regular granuloma look like at early stages of a tuberculosis infection.
Okay, some takeaway points from our talk. Assuming a time-dependent segregation rate, we're able to find new limits on replication and death rates than previously published by Gill et al. Specifically, as S increases throughout infection, we find lower limits on R and D. This implies we can find lower and upper bounds for these parameters. Bacteria must die in Mycobacterium tuberculosis infection in mice. That's the only way we can explain the data with our model. The replication death rates do not necessarily have to be explained with 4 intervals. We can still explain the data with 3 replication rates and 2 death rates by splitting into an early stage and late stages of infection. We're able to explain the data with quiescence or dormancy. Up to 2.5% of bacteria may quiesce at each cycle. This will lead to approximately 30% of the bacteria being quiesced at day 111. We can use a simple mechanistic model that models immune response, macrophages, infected macrophages, and bacteria, and we're able to explain the data with a necrosis modeled by a logistic function. We're able to show granuloma formation using CompuCell 3D with the assumptions previously shown. Future work, the mechanistic model isn't complete. Although we've built this mechanistic model and have gotten decent curve fits, we can still make it a little bit more robust. We can include antibiotic treatments and perhaps extend this model not only to mouse infections but to human infections as well, as potential for clinical treatment. We'd like to thank NIMBioS for funding this SRE program. We'd like to thank Dr. Sterner and Dr. Linhart for putting together all the events for this SRE program and for being excellent support. We'd like to give a big shout out to Dr. Ada and Dr. Ganisov for being amazing mentors and providing academic support.